ladies and gentlemen. On behalf of Dan Cromlin and myself, we have the opportunity to welcome here in this meeting and the topic of global clinic experience with non-biological complex drugs and their follow-on versions. Are we closer to the regulatory alignment? Therapeutic equivalence is the challenge, and it is for that kind of drugs like liposomes, glutamate, iron carbohydrates, or ocular emulsions. Regulatory approvals have been different in different areas, and I'm happy to have today people from different areas of the world, from regulatory area, but as well specialists in the field. We will have the 13 minutes time, which is intended for the speakers mainly, with 2 minutes at the end for urgent questions, but we will have by the very end a discussion where we are looking forward. So I will not go too much into the presentation of the speakers. You have all of that in the program, but we'll give the floor first now to Jan de Vlieger, who will as well present the non-biological complex drug working as we see on the first slide. Jan,
thank you very much, Stefan. Does the microphone work? Yes, wonderful. So indeed, I'm here discussing the non-biological complex drugs working group. I work at a foundation based in the Netherlands, Lygature. And what we do is we coordinate public-private partnerships. And we do that to pioneer new solutions for the benefit of patients. We work in 5 areas: in the neglected tropical diseases, in an area of sharing resources, regulatory innovation, patient engagement, and data infrastructure. So the NBCD working group is positioned in our regulatory innovation portfolio, and that's where I will focus my talk on for the next 10 minutes. So the working group has the mission to ensure that appropriate science-based approval and post-approval standards are created and globally introduced for NBCDs. And we have a partnership from public and private constituencies in the US and in Europe, and the foundation I work for is hosting this group for the last 7 years. So back in 2009, when Lygature was still Top Institute Pharma, we had a brainstorm session on the bioequivalence of complex drugs. It's quite some time ago. Based on those discussions, we issued a paper in 2011, and that actually kind of kick-started the discussion. So what are complex drug products? What is bioequivalence for these type of products, and what do we do? And since then, a number of key achievements came up, not only from the working group, but also from the partners we work with in terms of papers. We realized that, in fact, we all tried to speak English. The words of terminology in the area of science is challenging. So we defined a terminology list of complex issues, but we believe we should be on the same page. So what is equivalence? What is similarity? What is substitutability of a drug? Interchangeability. Do we understand what it is, and do we have the same vision of those? A few years ago, we published a book on NBCDs, which is kind of a reference material currently. And a key meeting occurred in 2016 in the New York Academy of Sciences. And based on that meeting, quite a number of events happened, and I will discuss a few and then leave the floor for the other speakers to discuss these things. So to give you an example, what are NBCDs? And the chair already mentioned a few. Before going into a formal definition of an NBCD, just some examples. This audience is very familiar with the liposomes, doxorubicin liposomes. There are also other NBCDs, such as the cyclosporine ophthalmic emulsions, iron sucroses, iron carbohydrate colloidal solutions, and sevelamer carbamate. So that's a phosphate binder that is a polymeric product. And another recent example, which we have seen in regulatory reviews, is glutarimer acetate and its generics or follow-ons. So those are the type of products we're talking about. And if you look at these products, there are lots of commonalities, but there are also lots of differences. And if you are trying to define a list of criteria where these products should meet, you would come up with a definition that is a bit like this. So an NBCD is not a biological product, and that is important to realize. So it's a synthetic medicinal product, and the active substance is not homomolecular. So it's often a mixture of active substances and which are closely related. And the reason why CLINAM is so important to us is that many of these products are nanomaterials, and they often contain nanoparticular structures. The key attribute of these products is that they cannot be fully characterized. You can characterize them up to a very large extent. Some products you can almost fully characterize, but most of them you cannot fully characterize. And that provides challenges for the scientists trying to characterize their products, but also for the regulators to see, is this product, if you're developing a generic or a follow-on, actually the same as the originator. And for all these products, as for instance, biologics and biosimilars, the manufacturing process is fundamental to ensure quality, safety, and efficacy. So how do these NBCDs compare to other drugs? If you look at the left-hand side of this graph, and I hope you can read it from the back, these are the small molecule drugs, which we are all used to. So they're rather small molecular weights. You can easily characterize them. They're manufactured through chemical synthesis. They're quite stable, and we know them quite well. So you can really make identical copies of these products. If you look at the complex drugs, both the biologics and the non-biologics, they share some commonalities. So they're complex, they're heterogeneous, and the manufacturing process is key. Manufacturing is either by living cells or organisms for biologics, or synthetic technologies, including nanotech for NBCDs. There are some challenges in immunogenicity, differences for biologics and NBCDs. However, for both of them, it's quite challenging to ensure identical copy versions. And that makes it a challenge for regulatory authorities to regulate these products. And we will hear some of the challenges and some of the solutions that our agencies have come up with for these type of products in the next talks. So some examples of what happened over the last few years with some of these products, without going into much detail, because I know next speakers will do so. So this is doxyl calyx, the doxorubicin liposome. We had different reviews in Europe and FDA, leading to approval in the US and rejection as a generic in Europe. Glatiramer acetate, Copaxone, more recent product, where I think Marisa will talk about as well, is approved as a generic in the US without clinical studies, and within Europe through a hybrid application, including clinical studies, approved also. It's a different product. It's not the same product that was reviewed. However, it's the same type of product in the same family. So that's an interesting approach of two regulators that have taken a different way of evaluating these. So there are many more of these products. And this is a complex slide, but what it shows you is that the complexity of determining therapeutic equivalence, as Stefan was saying, relies on bioequivalence measurements, determination, and pharmaceutical equivalence. So if one of these two is rather difficult, you can kind of still get away with evaluating. But if you combine these two challenges in one product, then you can imagine that reviewing applications for this or designing a pathway towards development is a challenging aspect. So in blue, you would see the NBCDs. In green, you would see the biologics. And there are two products here that are differently regulated in the world. So blue means NBCD either in Europe, where the green line means it's not an NBCD in the US or the other way around. So even the different regulators approach these products in a different way. And that is something where we believe alignment is possible. So also developers of generic similar products know how to move forward. So are there only challenges? No, there's lots of challenges, but there's also very good to mention advances in the regulation of these type of products. So this is a paper published based on the New York meeting I referred to, and we identified lots of progress being made all over the world. And we also identified the way forward. What are still the open topics that we need to discuss as a community to make next steps? So some of the unresolved challenges are listed here, and that's about critical quality attributes. to assess therapeutic equivalence of complex drug follow-on versions. How to provide the science base for these critical quality attributes. And I think Scott will discuss a few of these things here. What about nomenclature and labeling? Do we have a similar understanding of what we actually mean when we discuss follow-ons, similars, generics? And then again, we have seen in Europe, outside Europe, the approval of some of the follow-on products that are actually substandard. Do our pharmacovigilance systems allow us to pick those signals out? And what do we do? When do we decide this is enough? This product is really substandard or should have been regulated in a different way? What type of action do we take? So what do we believe that we need to do to advance the field to actually see alignment of science-based regulatory views worldwide? Of course, it's all about technologies, and that's why CLINAM is important, to see new technologies being able to analyze these type of complex products. And that's a technology development that the providers should help us with. It's really, really important that we engage all stakeholders, regulators, industry, academia, patients, to make sure that the steps that we are doing are meeting the requirements of the people that actually use the products later on. The challenge of correlating in vitro experience to in vivo outcome, that is also very important, because that will allow you to say, well, this is where we need clinical data and this is where we can rely on in vivo, in vitro correlations. And of course, it's important that we all know what we're talking about in terms of data. So please, innovators, generics, academia, publish your findings so we can discuss that in a transparent and open way. And also a call to regulators. We know you are bound to confidentiality, but you have lots of data that actually will help the community to solve these challenges that we've seen. And then, of course, the clinical relevant parameters, the critical quality attributes. So before going to next presentations, I want to highlight two sessions, two meetings. Next week, AAPS Guidance Forum, where we will discuss, together with FDA and the participants, their draft guidance released, and I'm sure Catherine will address this guidance later today. This is an important meeting where you can provide input, and we will hopefully make next steps in solving some of the challenges listed here around CQAs. And there's another meeting in November in Budapest, the third International Symposium on Scientific and Regulatory Advances in Biological and Non-Biological Complex Drugs, November 12 to 14. Thanks for that. I think I'm perfectly in time to take one critical question before the discussions. Thank you.
Thank you very much.